The innate immune system, also known as non-specific immune system and first line of defense, is a subsystem of the overall immune system that comprises the cells and mechanisms that defend the host from infection by other organisms in a non-specific manner. This means that the cells of the innate system recognize and respond to pathogens in a generic way, but, unlike the adaptive immune system, it does not confer long-lasting or protective immunity to the host. Innate immune systems provide immediate defense against infection, and are found in all classes of plant and animal life. They include both humoral immunity components and cell-mediated immunity components. The innate immune system is an evolutionarily older defense strategy, and is the dominant immune system found in plants, fungi, insects, and primitive multicellular organisms. The major functions of the vertebrate innate immune system include recruiting immune cells to sites of infection, through the production of chemical factors, including specialized chemical mediators, called cytokines, activation of the complement cascade to identify bacteria, activate cells and to promote clearance of antibody complexes or dead cells, the identification and removal of foreign substances present in organs, tissues, the blood and lymph, by specialized white blood cells. Activation of the adaptive immune system through a process known as antigen presentation, acting as a physical and chemical barrier to infectious agents. Anatomical barriers include physical, chemical and biological barriers. The epithelial surfaces form a physical barrier that is impermeable to most infectious agents, acting as the first line of defense against invading organisms. Desquamation of skin epithelium also helps remove bacteria and other infectious agents that have adhered to the epithelial surfaces. Lack of blood vessels and inability of the epidermis to retain moisture, presence of sebaceous glands in the dermis provides an environment unsuitable for the survival of microbes. In the gastrointestinal and respiratory tract, movement due to peristalsis or cilia, respectively, helps remove infectious agents. Also, mucus traps infectious agents. The gut flora can prevent the colonization of pathogenic bacteria by secreting toxic substances or by competing with pathogenic bacteria for nutrients or attachment to cell surfaces. The flushing action of tears and saliva helps prevent infection of the eyes and mouth. Inflammation. Inflammation is one of the first responses of the immune system to infection or irritation. Inflammation is stimulated by chemical factors released by injured cells and serves to establish a physical barrier against the spread of infection, and to promote healing of any damaged tissue following the clearance of pathogens. The process of acute inflammation is initiated by cells already present in all tissues, mainly resident macrophages, dendritic cells, histiocytes, Kupffer cells, and mastocytes. These cells present receptors, contained on the surface or within the cell, named pattern recognition receptors, which recognize molecules that are broadly shared by pathogens but distinguishable from host molecules, collectively referred to as pathogen-associated molecular patterns. At the onset of an infection, burn, or other injuries, these cells undergo activation and release inflammatory mediators responsible for the clinical signs of inflammation. Chemical factors produced during inflammation sensitize pain receptors, cause vasodilation of the blood vessels at the scene, and attract phagocytes, especially neutrophils. Neutrophils then trigger other parts of the immune system by releasing factors that summon other leukocytes and lymphocytes. Cytokines produced by macrophages and other cells of the innate immune system mediate the inflammatory response. These cytokines include TNF, HMGB1, and IL-1. The inflammatory response is characterized by the following symptoms: redness, heat, swelling, pain, possible dysfunction of the organs or tissues involved. Complement system. The complement system is a biochemical cascade of the immune system that helps, or a Euro-E complement seguro, the ability of antibodies to clear pathogens or mark them for destruction by other cells. The cascade is composed of many plasma proteins, synthesized in the liver, primarily by hepatocytes. The proteins work together to trigger the recruitment of inflammatory cells, tag pathogens for destruction by other cells by opsonizing, or coating, the surface of the pathogen, form holes in the plasma membrane of the pathogen, resulting in cytolysis of the pathogen cell, causing the death of the pathogen, rid the body of neutralized antigen-antibody complexes. Elements of the complement cascade can be found in many non-mammalian species including plants, birds, fish, and some species of invertebrates. Cells of the innate immune response. All white blood cells are known as leukocytes. Leukocytes are different from other cells of the body in that they are not tightly associated with a particular organ or tissue. Thus, they function similar to independent, single-cell organisms. Leukocytes are able to move freely and interact with and capture cellular debris, foreign particles, or invading microorganisms. Unlike many other cells in the body, most innate immune leukocytes cannot divide or reproduce on their own, but are the products of multipotent hematopoietic stem cells present in the bone marrow. The innate leukocytes include natural killer cells, mast cells, eosinophils, basophils, and the phagocytic cells including macrophages, neutrophils, and dendritic cells, and function within the immune system by identifying and eliminating pathogens that might cause infection. Mast cells. Mast cells are a type of innate immune cell that reside in connective tissue and in the mucous membranes. They are intimately associated with wound healing and defense against pathogens, but are also often associated with allergy and anaphylaxis. When activated, mast cells rapidly release characteristic granules, rich in histamine and heparin, along with various hormonal mediators, and chemokines, or chemotactic cytokines into the environment. Histamine dilates blood vessels, causing the characteristic signs of inflammation, and recruits neutrophils and macrophages. Phagocytes. The word phagocyte literally means eating cell. These are immune cells that engulf, that is phagocytous, pathogens or particles. To engulf a particle or pathogen, a phagocyte extends portions of its plasma membrane, wrapping the membrane around the particle until it is enveloped. Once inside the cell, the invading pathogen is contained inside an endosome, which merges with a lysosome. The lysosome contains enzymes and acids that kill and digest the particle or organism. In general, phagocytes patrol the body searching for pathogens, but are also able to react to a group of highly specialized molecular signals produced by other cells, called cytokines. The phagocytic cells of the immune system include macrophages, neutrophils, and dendritic cells. Phagocytosis of the host's unregistered trademark-owned cells is common as part of regular tissue development and maintenance. When host cells die either internally induced by processes involving programmed cell death or caused by cell injury due to a bacterial or viral infection, phagocytic cells are responsible for their removal from the affected site. By helping to remove dead cells preceding growth and development of new healthy cells, phagocytosis is an important part of the healing process following tissue injury. Macrophages. Macrophages, from the Greek, meaning large eaters, are large phagocytic leukocytes, which are able to move outside of the vascular system by moving across the walls of capillary vessels and entering the areas between cells in pursuit of invading pathogens. In tissues, organ-specific macrophages are differentiated from phagocytic cells present in the blood called monocytes. Macrophages are the most efficient phagocytes, and can phagocytose substantial numbers of bacteria or other cells or microbes. The binding of bacterial molecules to receptors on the surface of a macrophage triggers it to engulf and destroy the bacteria through the generation of AAUO or respiratory burst AUO, causing the release of reactive oxygen species. Pathogens also stimulate the macrophage to produce chemokines, which summons other cells to the site of infection. Neutrophils. Neutrophils, along with two other cell types, eosinophils and basophils, are known as granulocytes due to the presence of granules in their cytoplasm, or as polymorphonuclear cells due to their distinctive lobe nuclei. Neutrophil granules contain a variety of toxic substances that kill or inhibit growth of bacteria and fungi. Similar to macrophages, neutrophils attack pathogens by activating a respiratory burst. The main products of the neutrophil respiratory burst are strong oxidizing agents including hydrogen peroxide, free oxygen radicals and hypochlorite. Neutrophils are the most abundant type of phagocyte, normally representing 50 to 60% of the total circulating leukocytes, and are usually the first cells to arrive at the site of an infection. The bone marrow of a normal healthy adult produces more than 100 billion neutrophils per day, and more than 10 times that many per day during acute inflammation. Dendritic cells. Dendritic cells are phagocytic cells present in tissues that are in contact with the external environment, mainly the skin, and the inner mucosal lining of the nose, lungs, stomach, and intestines. They are named for their resemblance to neuronal dendrites, but dendritic cells are not connected to the nervous system. Dendritic cells are very important in the process of antigen presentation, and serve as a link between the innate and adaptive immune systems. Basophils and eosinophils. Basophils and eosinophils are cells related to the neutrophil. When activated by a pathogen encounter, basophils releasing histamine are important in defense against parasites, and play a role in allergic reactions. Upon activation, eosinophils secrete a range of highly toxic proteins and free radicals that are highly effective in killing bacteria and parasites, but are also responsible for tissue damage occurring during allergic reactions. Activation and toxin release by eosinophils is, therefore, tightly regulated to prevent any inappropriate tissue destruction. Natural killer cells. Natural killer cells, or NK cells, are a component of the innate immune system that does not directly attack invading microbes. Rather, NK cells destroy compromised host cells, such as tumor cells or virus-infected cells, recognizing such cells by a condition known as missing self. This term describes cells with low levels of a cell surface marker called MHCI, a situation that can arise in viral infections of host cells. They were named natural killer because of the initial notion that they do not require activation in order to kill cells that are missing self. For many years, it was unclear how NK cell recognized tumor cells and infected cells. It is now known that the MHC makeup on the surface of those cells is altered and the NK cells become activated through recognition of missing self. Normal body cells are not recognized and attacked by NK cells because they express intact self-MHC antigens. Those MHC antigens are recognized by killer cell immunoglobulin receptors that, in essence, put the brakes on NK cells. The NK92 cell line does not express KIR and is developed for tumor therapy. I-cubed T cells. Like other unconventional T-cell subsets bearing invariant T-cell receptors, such as CD1d-restricted natural killer T cells, I-cubed T cells exhibit characteristics that place them at the border between innate and adaptive immunity. On one hand, I-cubed T cells may be considered a component of adaptive immunity in that they rearrange TCR genes to produce junctional diversity and develop a memory phenotype. However, the various subsets may also be considered part of the innate immune system where a restricted TCR-NK receptors may be used as a pattern recognition receptor. For example, according to this paradigm, large numbers of Vi-cubed 9/Vi2 T cells respond within hours to common molecules produced by microbes, and highly restricted intraepithelial Vi-1 T cells will respond to stressed epithelial cells. Other vertebrate mechanisms. The coagulation system overlaps with the immune system. Some products of the coagulation system can contribute to the non-specific defenses by their ability to increase vascular permeability and act as chemotactic agents for phagocytic cells. In addition, some of the products of the coagulation system are directly antimicrobial. For example, beta-lysine, a protein produced by platelets during coagulation, can cause lysis of many gram-positive bacteria by acting as a cationic detergent. Many acute-phase proteins of inflammation are involved in the coagulation system. Also, increased levels of lactoferrin and transferrin inhibit bacterial growth by binding iron, an essential nutrient for bacteria. Neural regulation of innate immunity. The innate immune response to infectious and sterile injury is modulated by neural circuits that control cytokine production. The inflammatory reflex is a prototypical neural circuit that controls cytokine production in spleen. Action potentials transmitted via the vagus nerve to spleen mediate the release of acetylcholine, the neurotransmitter that inhibits cytokine release by interacting with alpha-7 nicotinic acetylcholine receptors expressed on cytokine-producing cells. The motor arc of the inflammatory reflex is termed the cholinergic anti-inflammatory pathway. Pathogen specificity: the parts of the innate immune system have different specificity for different pathogens. Innate immune evasion: cells of the innate immune system, in effect, prevent free growth of bacteria within the body. However, many pathogens have evolved mechanisms allowing them to evade the innate immune system. Evasion strategies that circumvent the innate immune system include intracellular replication, such as in Mycobacterium tuberculosis, or a protective capsule that prevents lysis by complement and by phagocytes, as in Salmonella. Bacteroid species are normally mutualistic bacteria, making up a substantial portion of the mammalian gastrointestinal flora. Some species are opportunistic pathogens, causing infections of the peritoneal cavity. These species evade the immune system through inhibition of phagocytosis by affecting the receptors that phagocytes use to engulf bacteria or by mimicking host cells so that the immune system does not recognize them as foreign. Staphylococcus aureus inhibits the ability of the phagocyte to respond to chemokine signals. Other organisms such as M. tuberculosis, Streptococcus pyogenes, and Bacillus anthracis utilize mechanisms that directly kill the phagocyte. Bacteria and fungi may also form complex biofilms, providing protection from the cells and proteins of the immune system. Recent studies indicate that such biofilms are present in many successful infections, including the chronic Pseudomonas aeruginosa and Burkholderia sanocypacia infections characteristic of cystic fibrosis. Evasion of the innate immune system by virus. In essence, antiviral host defense and creation of an effective antiviral state in a cell is due to the role played by type I interferons, secreted mainly by dendritic cells. Viral components are recognized by different receptors. Toll-like receptor are located in the endosomal membrane and recognizes double-strand RNA, MDA5 and or IgIR in the cytoplasm and recognize respectively long dsRNA and phosphate-containing dsRNA. The viral recognition by MDA5 and or IgI in the cytoplasm induces a conformational change between the caspase recruitment domain and the card-containing adapter MAVS. In parallel, the viral recognition by TLR3 in the endocytic compartments induces the activation of the adapter protein TRIF. These two pathways converge in the recruitment and activation of the chymicron/TBK1 complex, inducing phosphorylation and homo- and heterodimerization of transcription factors IRF3 and IRF7. These molecules are translocated in the nucleus, where they induce IFN production with the presence of CJUN and ATF2. Then, IFN bind to the IFN receptors and induce the expression of hundreds of interferon-stimulated genes by the JAK-STAT pathway. These ISG produce proteins with antiviral properties, like protein kinase R, which inhibits viral protein synthesis, or the 2A2, 5A2 oligoadenylate synthetase family, which degrade viral RNAM. These molecules establish an effective antiviral state in the cell. Some viruses are able to evade this immune system, by producing molecules that interfere with IFN production pathway. For example, the influenza A virus NS1 protein binds single-strand and or dsRNA, inhibiting type I IFN production. Later in the defense reaction, this virus blocks PKR activation and the establishment of the antiviral state. The dengue virus also inhibits type I IFN production by blocking IRF3 phosphorylation using Nova Scotia 2b3 protease complex. Innate immunity in other species. Host defense in prokaryotes, bacteria, utilize a unique defense mechanism, called the restriction modification system to protect themselves from pathogens, such as bacteriophages. In this system, bacteria produce enzymes, called restriction endonucleases, that attack and destroy specific regions of the viral DNA of invading bacteriophages. Methylation of the host's own DNA marks it as self, and prevents it from being attacked by endonucleases. Restriction endonucleases and the restriction modification system exist exclusively in prokaryotes. Host defense in invertebrates. Invertebrates do not possess lymphocytes or an antibody-based humoral immune system, and it is likely that a multi-component, adaptive immune system arose with the first vertebrates. Nevertheless, invertebrates possess mechanisms that appear to be precursors of these aspects of vertebrate immunity. Pattern recognition receptors are proteins used by nearly all organisms to identify molecules associated with microbial pathogens. Toll-like receptors are a major class of pattern recognition receptor that exists in all chelamates, including humans. The complement system, as discussed above, is a biochemical cascade of the immune system that helps clear pathogens from an organism, and exists in most forms of life. Some invertebrates, including various insects, crabs, and worms, utilize a modified form of the complement response known as the prophenyloxidase system. Antimicrobial peptides are an evolutionarily conserved component of the innate immune response found among all classes of life and represent the main form of invertebrate systemic immunity. Several species of insect produce antimicrobial peptides known as defensins and secropins. Proteolytic cascades. In invertebrates, pattern recognition proteins trigger proteolytic cascades that degrade proteins and control many of the mechanisms of the innate immune system of invertebrates, including hemolymph coagulation and myelinization. Proteolytic cascades are important components of the invertebrate immune system because they are turned on more rapidly than other innate immune reactions because they do not rely on gene changes. Proteolytic cascades have been found to function the same in both vertebrate and invertebrates, even though different proteins are used throughout the cascades. Clotting mechanisms. In the hemolymph, which makes up the fluid in the circulatory system of arthropods, a gel-like fluid surrounds pathogen invaders, similar to the way blood does in other animals. There are various different proteins and mechanisms that are involved in invertebrate clotting. In crustaceans, transgalactaminase from blood cells and mobile plasma proteins make up the clotting system, where the transgalactaminase polymerizes 210 kDa subunits of a plasma clotting protein. On the other hand, in the horseshoe crab species clotting system, components of proteolytic cascades are stored as inactive forms in granules of hemocytes, which are released when foreign molecules, like lipopolysaccharides, enter. Host defense in plants. Members of every class of pathogen that infect humans also infect plants. Although the exact pathogenic species vary with the infected species, bacteria, fungi, viruses, nematodes, and insects can all cause plant disease. As with animals, plants attacked by insects or other pathogens use a set of complex metabolic responses that lead to the formation of defensive chemical compounds that fight infection or make the plant less attractive to insects and other herbivores. Like invertebrates, plants neither generate antibody or T-cell responses nor possess mobile cells that detect and attack pathogens. In addition, in case of infection, parts of some plants are treated as disposable and replaceable, in ways that very few animals are able to do. Walling off or discarding a part of a plant helps stop spread of an infection. Most plant immune responses involve systemic chemical signals sent throughout a plant. Plants use pattern recognition receptors to recognize conserved microbial signatures. This recognition triggers an immune response. The first plant receptors of conserved microbial signatures were identified in rice and in Arabidopsis. Plants also carry immune receptors that recognize highly variable pathogen effectors. These include the NBSLR class of proteins. When a part of a plant becomes infected with a microbial or viral pathogen, in case of an incompatible interaction triggered by specific elicitors, the plant produces a localized hypersensitive response, in which cells at the site of infection undergo rapid programmed cell death to prevent the spread of the disease to other parts of the plant. HR has some similarities to animal pyroptosis, such as a requirement of caspase-1-like proteolytic activity of VPEI cubed, a cystine protease that regulates cell disassembly during cell death. Resistance proteins, encoded by genes, are widely present in plants and detect pathogens. These proteins contain domains similar to the NOD-like receptors and Toll-like receptors utilized in animal innate immunity. Systemic acquired resistance is a type of defensive response that renders the entire plant resistant to a broad spectrum of infectious agents. SAR involves the production of chemical messengers, such as salicylic acid or jasmonic acid. Some of these travel through the plant and signal other cells to produce defensive compounds to protect uninfected parts, for example, leaves. Salicylic acid itself, although indispensable for expression of SAR, is not the translocated signal responsible for the systemic response. Recent evidence indicates a role for jasmonates in transmission of the signal to distal portions of the plant. RNA silencing mechanisms are also important in the plant's systemic response, as they can block virus replication. The jasmonic acid response is stimulated in leaves damaged by insects, and involves the production of methyl jasmonate. See also, apoptosis, Toll-like receptor, innate lymphoid cell, references. Akira, S. Uematsu, and Otakeuchi. 2006. Pathogen Recognition and Innate Immunity. Cell 124 783 801 Baum, A., and A. Garcia-Sasta. 2010. Induction of type I interferon by RNA viruses, cellular receptors and their substrates. Amino acids 381283 1299. Garcia-Sasta, A., Agarov, D. Matasov, S. Brandt, D. E. Levy, J. E. Durbin, P. Police, and T. Muster. 1998. Influenza A virus lacking the NS1 gene replicates in interferon-deficient systems. Virology 252 324 330, Le Bon, A., and D. F. Tough. 2002. Links between innate and adaptive immunity via type I interferon. Kerr. Open. Immunol 14 432 436. Liu, Y. M., J. Fornick, N. Crochet, G. Bajwa, O. Peritiseri, L. Martinez Subredo, S. Akira, M. A. Gill, A. Garcia-Sasta, M. G. Katz, and M. Gale, Jr. 2008. Distinct RIGI and MDA5 signaling by RNA viruses in innate immunity. J. Viral. 82 335 345. External links: the Center for Modeling Immunity to Enteric Pathogens.